Okay, thank you, Thomas. Thank you very much for the invitation to this great symposium. So yes, I'm going to tell you about active flow in the three-dimensional cell aggregate. So I'm a theorist. I'm working closely with developmental biologists, essentially. Those are essentially on tissue morphogenesis. And this is some examples of my recent interest in morphogen gradient. I'm interested in the mechanics of epithelial flow. This is a movie of the histoblast nest. On which we are working with Nick Tapon. I'm also interested on the theory side on the physics of polar and nematic active surfaces. We have done a recent study on the shapes of these surfaces. And we have a preprint on mechanochemical pattern formation with Jean-Paul Vincent, which is on the formation of the veins in the Drosophila wing. But today I'm not going to talk about that. I'm going to talk about 3D cell aggregate. And to explain what we have been doing recently, I would like to first go back in the past and explain what we had done before so that it's clear where we're coming from. And one thing we have done is, together with Eva Palush, was at the time to look at single-cell migration. This is a movie of a single cell moving in a channel. And in these movies, one can image myosin distribution, which is mostly in the back of the cell. And also, if one looks at this movie, one can see that the myosin spots are not moving in the reference frame of the lab, which indicates that there is a retrograde flow in the reference frame of the cell from the front to the back of the cell. And us and others have looked at this kind of system by using active surface theory, where the idea is that there is a stress distribution on the surface of the cell, which has two contributions. This is the tension tensor on the cell surface. It describes the actomyosin network. One contribution is a viscous flow with the idea that this actin polymer network, they can, because they turn over, they can release elastic stresses, and so on long-term scale, they have an effective viscosity. And in addition, there is a surface tension, active surface tension, that comes from the action of myosin and the contractile stresses that they generate. And so the idea is simply that if you have a network and in some place you have more myosin molecular motor, this is going to generate a flow because of the higher contractility, and this is described in this theories by a gradient of active tension. And when one works out the force balance, one can see that this gradient of active tension creates a flow. And then this is an example of a solution of this model. This cell is actually calculated by putting a gradient of active tension in the back. Then there is a cortical flow, which is shown by the black dots, which move from the front to the back. And because of the friction between the cell and the channel, the cell is then moving forward. And one can make a prediction as a function of the friction coefficient of the cell velocity. At low friction, it vanishes because the cell cannot engage with the wall. When the friction is increasing, the velocity becomes significant. At the same time, the retrograde flow is decreasing, the retrograde flow in the reference frame of the lab, because if the friction is too high, the cortex cannot flow relative to the channel. And we could test this experimentally by changing the coating of the wall. And for coating giving rise to high friction, the cell is moving, but with very little retrograde flow in the reference frame of the lab. At low friction, the cell is not moving, but there is a retrograde flow, but it does not manage to propel the cell. And there are intermediate cases where at intermediate friction, the cell is moving, but there is still a retrograde flow which is visible. So that's one side of what we had been doing, which is on understanding the physics of the actomyosin cortex. On the other hand, we have done some work on the epithelial mechanics, working with a 3D vertex model. So vertex models are very famous, come in different flavors. The one that we used was a model for epithelial monolayers, where we represented the tissue by a set of vertices, apical vertices and basal vertices, and then the cells are defined by the vertices that belong to them. And the key aspect is that there are different surface tension on the interface of this, of the apical, basal, and lateral interfaces of these cells. Then one can simulate this. For instance, here that is a cylindrical tissue which is mechanically balanced, which means that the forces coming from this surface tension result in a net zero force on each of these vertex. Then we have used it to look at different situations. One interesting one, I think, is the one where we looked at the formation of pancreatic tumor in mouse. And so this is experiment. Those are experiments done in the lab of Axel Behrens, where This is an image of a tube inside the mouse pancreas, and they have transgenic mice in which they can induce tumors, and then one can look at the shape of these tumors inside the tube. And we wanted to understand why the tissue was deforming, and one key information that we noticed was that, so this is a cross-section of the tube, and that the myosin distribution was normally in the healthy tissue more intense on the apical side, but it was modified in the transformed cancerous cells, and the myosin was more homogeneous apically and basally, which indicated a redistribution of forces in the transformed cells and the healthy cells. And when we input this in our simulation, we saw this interesting behavior that in small tubes, the tumors were growing to the outside. In large tubes, they were growing to the inside. And the reason is that this difference of surface tension creates a spontaneous curvature, and which makes the tumor goes to the inside, but this only works mechanically if the tube is large enough. And then we tested this experimentally, and this is indeed what happens in a large pancreatic duct. The tumors are growing to the inside, otherwise they are growing to the outside. So starting from these two different approaches, we wanted to do something mixing them to have a model which would apply for tissues, but which would be better or more capturing more effects than the 3D vertex model by taking into account the mechanics and the physics of the actomyosin network. And that would then give us a physical description of cell aggregates. And so this is what Alejandro Torres Sanchez has done during his postdoc in my group. He has developed a model that we have called the interacting active surface model, and Alejandro is now a group leader at EMBL in Barcelona. So the idea of this model is it's a computational model. We describe each cell by a surface, so we only focus on cell surface mechanics. There is a flow, velocity field on the cell surface, which is going to be determined by force balance. On a curved surface, force balance takes this form. There is a covariant derivative of the tension tensor plus the external force, which has to vanish. To specify the model, one needs to say what these forces are. As far as the internal tension is concerned, we come back to our usual simple description of the surface of the cell as an active fluid, which has a viscous tension and an active tension that depends on the distribution of myosin molecular motors. For the external force, we have different types of external force. One is clearly the internal cell pressure, which is pushing on the cell surface, and in our case, we say comes from the fact that there is a conserved cell volume. And also, now what we are interested in is cell-cell interactions. And so here we describe cell-cell interaction in the, I think, most natural way. We said that there are linkers on the cell surface. If they are close enough, they can bind, and then they make an elastic link. And when it turns out that if one works out the physics of these linkers in the thermodynamic limit, where other deformation of the cell are slow compared to the diffusion of these linkers, this effectively gives rise to this kind of potential where each point on a cell is interacting with any other point on another cell. And this potential then takes a classic form. It's repulsive at short time scale. It has a preferred distance where the linkers are at their preferred distance. And then it disappears because eventually, if the cells are far away, they don't see each other. Okay, and so those are essentially the ingredients. And then a lot of the work is done in this slide because one has to start from this continuum mechanics of active surfaces and using a finite element method, turn it into equations for the deformation of the mesh, but also calculation of the flow on these surfaces. And so that's what Alejandro has done. And then we started by making sure that what we were doing made sense. And so we first made simulation of the simplest thing we could think about, something that is studied experimentally and theoretically, just putting two cells together and seeing when we increase the adhesion, what happens. And of course, the contact size increases. This we know there is a classic simplified description of this, where the steady-state shape is described by the Young-Dupré equation, which determines the contact angle between the cells based on their surface tension and based on how much adhesion energy there is at the contact. And this simplified theory gives a prediction for the size of the contact as a function of the cell adhesion strength. We took our simulation, we compared this to this prediction to make sure that it was okay. It's pretty good. And then if we decrease the bending modulus of our surfaces, it gets really good because then the shape near the contact point becomes sharper and sharper, and this is, and gets closer to the simplified Young-Dupré equation. So, okay, the other prediction of the analytical theory is that if the adhesion energy at the interface becomes too high, its effective tension becomes negative, and it becomes energetically favorable to increase its area, which means that it's not flat anymore, it's unstable. And this is indeed what we see in the simulation. We were curious to see what would happen next, and actually we get this S shape, which keeps deforming, and with the particular physical ingredients that we have in the simulation, it just never stops. It will eventually cross, this interface will eventually cross the other side of the cell. So if this was a real experiment, something would be missing, clearly. Okay, and then once we have done this, we go to simulations with more cells. Those are two examples. This is a case where we effectively made an epithelial monolayer by putting cells together in a plane. And then we imposed a gradient of active tension, so assuming in practice that there would be more myosin on one side than the other. And then we looked at the deformation, which, as one would expect from what happens, for instance, in apical constriction, leads to an overall bending of the tissue. But also, in principle, we can solve for the flow, for the cortical flow on the surface of each cell during this process. And another example of what we do here is to introduce cell division, and then we can simulate this growing aggregate. Here there's relatively basic physics, just uniform surface tension, so we get this relatively homogeneous looking aggregate. So we have developed this tool, and then we wanted to actually look at some real case. So, and this is the work of Quentin Vagne. So he has been looking at three-dimensional rotation of cell doublets. So the experiments that we looked at were done in the lab of Daniel Rivlin, and they looked at MDCK, which are embedded in 3D in matrigel, and the single cells are not doing much, but when they divide, the two daughter cells attach to each other, and then they start to rotate. Okay, that's what we see, an example here. And this kind of rotation has been described before on a two-dimensional substrate, but here it's in 3D. So we wanted to understand this, so we first have taken this movie and we have segmented them, and then we can start to quantify some things. One thing is that we can quantify the velocity of each cell, and from these velocities define a rotation vector, okay, whose direction gives the axis around which the doublet is rotating. And after cell division, if we look at the magnitude of the rotation, we see that the rotation increases and then saturates at a velocity of about 1 turn every 2 hours. We can also look at the orientation of the rotation axis. Because it's in 3D, it moves around. It should, because there's no reason for the axis to have one particular orientation. That also implies that this object is not chiral, so a rotating object is not necessarily chiral. This one is not chiral. One can see this because if I look at a doublet rotating from one side, I would see it rotating to the right, let's say. If I look at it from the other side, I would see it rotating to the left, which also means that if I look at a doublet in a mirror, I can still rotate it and get the same, map it onto the original doublet. Okay, and then we were also interested in looking at the shape of the interface between the cells. And so we see as the interface, after cell division, as the doublet rotates faster and faster, the interface also deforms more and more. And this is a map of the height of the interface relative to a reference plane. And we see that it has overall this Pringle shape, or this saddle node shape. So we thought, okay, we need to quantify this interface shape, see if we can learn something from it. And so with Quentin, we played a little bit around decomposing the interface shape on a circle. And we used polynomials to define a set of modes, a saddle node, a ball, a freefold, which is also called a monkey saddle, because a monkey could sit on it with its tail here, and a yin-yang mode, which we call like this, because if you have this deformation mode and you look at the doublet rotating, it looks like a yin-yang. The nice thing is that this mode, they have different symmetries. The saddle node is characterized by a nematic order. The ball is rotationally symmetric, so just a scalar. the monkey saddle is a free-fold mode with a free invariance, a profile with invariance by rotation of 2 pi over 3, and the yin-yang is equivalent to a polar mode. We are very happy about this, and then we realized that those are actually the Zernike polynomials, which were found in 1934. So we were scooped. So then we take our interface and we decompose it into modes. And we see that the saddle node is dominating, followed by ball, free-fold, and yin-yang mode. Okay, so we have characterized the shape and the rotation, and now the question is where does it come from? And so our collaborators looked at the distribution of E-cadherin, which is mostly at the cell interface, at actin, which is on the interface and on the cell, even if it's not visible here. And the interesting thing is they also look at myosin, and we saw that there was these myosin clusters which are located close to the cell-cell interface. So we thought, okay, we need to quantify what these clusters are doing. And so to do this, we define a polarity axis, so an arrow, which is going to point where there is more myosin in the cell. And then we can, from these polarity arrows, define angles. And so we have one angle which measures which is linked to the polarity vector and the axis joining the two cells. These angles are roughly 90 degrees. And there is a second angle if I look at the doublet from the top here, and I looked at this angle beta between the two polarity axes. We found roughly 180 degrees, so that means that the polarity angles are like this. They are opposite to each other, and 90 degrees from the cell-cell axis. So knowing this, we said, okay, we are going to come back to this interacting active surface model, and now we are going to take the doublet we had initially, but we will add this polarity axis, which is going to create a gradient of active tension. Okay, so there is a gradient of active surface tension, which is higher here and here. Then when we will solve our equation, we will find a cortical flow, a velocity on the cell surface. This doublet is embedded in matrix gel, so we assume that there is an external friction force that opposes the velocity, and we can see what happens. And this is what we observe for increasing value of this gradient of active surface tension. And so when we increase this gradient, the doublet rotates. It rotates in the same way that what we see in the experiment, so that's good. And also the interface deforms. In fact, if we increase too much the gradient of active tension, we see that the interface keeps deforming here. But here, for intermediate difference of active tension, it saturates. So then we can do some tests to see that this model makes sense. One simple test is to say we just remove suddenly the gradient of active tension, and of course the rotation stops. The interface relaxes, and this we can do in experiment. Our collaborators can do this in experiment with laser ablation, and indeed the doublet stops rotating, and then it starts to, after a while, the clusters reform, and then it starts to rotate again. But transiently, the cluster is formed. We can verify this quantitatively. Another more subtle experiment is to locally induce an additional excess of myosin. This is what we do computationally here. Here we suddenly create a secondary spot. And if we do that, we see that the doublet keeps rotating, but it moves away from the point of activation. And we can also do this using a MDCK optogenetic cell line. One can induce an excess of myosin at some point on the cell surface, and that indeed makes the doublet move away from the point of activation. This is a quantification of this for several doublets, measuring the position of the center of mass of the cell after optogenetic activation, and it moves away from the point of optogenetic activation. So that sounds good, but then we realized that we had something missing, which is that if we look now at the interface shape in simulation and we projected onto these four modes, we had a pure yin-yang, which was not in accordance with the experiment where we see these four different modes. So what is it that's missing? And so one first cue is that I showed you that these different modes, they have axes. They have nematic axis, free-fold axis, polar axis. And then we can ask if this axis, how correlated it is to the axis of rotation of the cell. One has to think a little bit about the symmetries to do this properly, but it's possible to do this correlation. And if we do that, we see that only the yin-yang deformation is correlated to the rotation, not the saddle node or the free-fold. So there is a saddle node deformation, but basically it's not oriented in a way that is consistent with the direction of rotation. So that gives us a clue that there is indeed a relation between yin-yang and rotation that we don't see for saddle node and freefold. But then what about the other modes? So one thing that we can do is we take these segmented cells, and then we take them and computationally we arrange them in a consistent way, so that here the neighboring cell is always pointing at the top, and one other axis which is turning here is the axis of rotation. Then we can pull all the data, put it together, project it on a sphere, and that gives us a map of the myosin distribution. There is no myosin here because there's no myosin at the cell-cell contact. Most of the myosin is actually at the ring, close to the cell-cell interface. This is the spot I've been talking about. But the distribution is a bit more complex. It's also maybe not very visible here, but there's actually a secondary weaker spot on the other side of the initial spot that I was describing. So now to make sense of this and how to explain the interface shape, we thought about using the Curie principle. So I did my PhD in Institut Curie, so I heard about the Curie principle there, which is that the symmetry of the causes are to be found in the effects. Or in our case, what this means is that, okay, if we see some broken symmetry in the effect, we have to find a broken symmetry in the causes. So how to do this in practice? So it's probably most people know that if you have molecules, you can characterize them according to their symmetry group. For instance, the molecule of water have an axis of rotation where it's invariant by rotation of pi, and it has two mirror symmetry planes, which leaves it invariant. And then physicists and chemists use this to make generic prediction about matter made of these molecules. Okay? And the fact that there are these particular elements of symmetry in the symmetry group says that this molecule belongs to this symmetry group in Schoefflitz notation C to V. So then we said, okay, we're going to do the same thing with the doublet. We're going to ask, depending on the distribution of myosin on the cell surface, what should we expect in terms of symmetry group, and what should we expect in terms of cell interface? So the cause is the distribution of myosin or cortical tension, and the effect is the shape of the interface. So for instance, if we have a doublet with two polarity axes pointing towards each other, then no deformation of the shape of the interface is compatible with the symmetries of this object. And if we do a simulation, indeed it's flat. If we take two shifted polarity vectors like we had before, this is the symmetry group C2H. A freefold and a yin-yang are compatible with this symmetry group. In the simulation, we observed only a yin-yang and a rotation, but then this tells us this had to be because a saddle node and ball could not emerge. You could also have a situation where you have one cell with a higher average tension than, or myosin distribution than the other. That's another symmetry group, which can only create a ball mode. This is what happens in simulation. Or you can make a simulation where you have two spots in one cell. That's another symmetry group, C2V, and this one can generate the saddle node, and indeed this is what it would do. So how do we use this? Well, then we can take the data and see if indeed we see this relationship between the distribution of myosin and these different deformation modes. And so, for instance, we can take the average intensity of myosin in one cell, take the difference with the second cell, and see if it correlates with the ball mode. And indeed, we see a correlation. What this means simply is that the cell with higher tension is pushing towards the cell with lower tension. And we can also do the same thing with the saddle node, where we take the difference. So it's a little bit more complicated to do, but we look at the nematic order of the myosin distribution in one cell. In the second cell, take the difference and correlate it with the orientation of the nematic of the saddle node. And if we do this, we do find a positive correlation. So that supports the idea that this more complex distribution of myosin, where we had two opposite spots, creates the saddle node. And so that's basically my conclusion for this part, which is that the Curie principle allows us to decompose the cause and effect and understand this complex shape deformation process by connecting it to the organization of the myosin on the cell surface. And this is a proof of principle of simulation where we integrated myosin distribution combining these different modes to generate a simulation which has the same decomposition as in the experiment. Okay. Now, in the last part, I would like to tell you about what we have been doing on larger aggregates and the data analysis that we have done on rosette formation in three-dimensional stem cell aggregate. And this is the work of Matthew Smith and Max Kerwinter. So, a few years ago, the lab of Magdalena Zernik-Jagetz has shown that one can take stem cells and then grow them in vitro, embryonic stem cells, and they will spontaneously form this kind of structure, so which is basically a spherical monolayer with a lumen at the center. And they proposed that this was mimicking the formation of the lumen in the epiblast in mouse embryos. And so they call this self-organization because just from a single cell, by dividing and rearranging, it was capable of making this structure. They also showed that following a beta-1 integrin knockout, the cell lost their capacity to read a signal from the extracellular matrix, which led to a disorganization, and then they make this clump of cells which does not look anymore like these nicely ordered structures. So what we did there is we asked them to take some movies for us of this process. And in the interest of time, I'm not showing the original data, just directly the segmentation. And the segmentation, we have done it using deep learning tools and a Fiji plugin based on active mesh deformation, which we have published recently. So we segment the cells of this movie, which lasts for about 30 hours and go to about 8 cells. And we can see that, indeed, there is already at these early stages a difference between the wild type and beta-1 integrin knockout. So the wild type is forming a nice spherical structure, where roughly the cells have a common contact point in the center, even if we cannot really see it here because it's hidden. Whereas in the beta-1 integrin knockout case, we make this much more disordered structure. So we wanted to quantify this. For this, we take the center of each cell, we take the distance of the center of the cell to the center of the aggregate, and then we define an order parameter, which is the coefficient of variation, so is the standard deviation of this distance divided by the mean. Then we take our movies, and we time align them. We choose a common reference point. Time equals 0 is the time at which the aggregate goes from 2 to 3 cells. Okay, and because we have looked at the cell cycle time, we know that it's about 11 hours, and then we know that over this time we have roughly 1, 2, 4, 8 cells. And then as a function of time, the blue curve describes the wild type, and the red curve the beta-1 integrin knockout. And what we see is that indeed already at these early stages there is a difference of organization. The wild type is more ordered, especially once the aggregate has 8 cells, than the beta-1 integrin knockout. So we wanted to look at these movies to see what we could understand about this difference and the mechanism for rearrangement of an aggregate like this into an ordered structure. The other thing that is visible in the movie is that the cells are moving quite a lot. They are moving mostly along the surface of the aggregate. And we can calculate the cell velocity as a function of time. We see that it tends to increase over time as the aggregate gets bigger. And one thing also we were wondering about is that is this aggregate rotating as a solid object or not? And so we can calculate a quantity which describes the rotation, solid rotation contribution of this aggregate. And so it exists, there is a non-zero contribution. But if we remove the solid rotation from the velocity of the cells, we get the green curve here, which is a little bit lower than the blue curve, but not much lower, which means that the cells are still have a significant motion relative to each other inside the aggregate. Okay, so where does the rotation come from? So we looked at movies where the cells are labeled with E-cadherin and actin. Here we'll only show E-cadherin. So this movie here is now obtained by taking the cells and then computationally displacing them from the center so that one can see what's going on. And also on the cells, we have mapped the E-cadherin intensity. Whenever we see an interface, we divide the E-cadherin signal by the amount of cells participating to the interface so that the signal is really the signal of E-cadherin per cell. Okay, and what we can see here from the movie is that there is a lot of myosin on the contour of the outer boundary of the cell, and also on the inside. So this is maybe not very visible, but it's going to be more visible here. Those are cells now that are taken. They are rotated so that they face us the same way that they face the center of the aggregate. And so in the wild type, we see all these spots here that form near the center, which are lost in the beta-1 integrin knockout. So clearly there is a strong difference here. We can also then look at the other interface of on the outer interface of the cells. We use the velocity axis to orient them coherently. And what one can also guess from this is that there tends to be more cadherin in the back of the cell than in the front of the cell. And this can be verified by, again, doing the same thing I was showing before, taking all the cells, arrange them in a coherent way, in that case, so that the blue arrow is pointing to the center of the aggregate, and that the green arrow is the common velocity axis of the cells. Then we map all the E-cadherin intensity, and we get this profile. We have indeed this spot at which is pointing to the inside. And we also have this ring here, which is clearly polarized. There is an excess of E-cadherin in the back of the cell compared to the front. Okay? And we have other ways to check that the E-cadherin distribution near the outer cell surface has a polarity which is opposite to the velocity. So briefly, so we then wanted to understand a little bit the link between this and how the aggregate organizes. And so I'm going to show very briefly a simple model, a polarity-based model, where the idea is that each cell has two polarity axes. One which is pointing to this little E-cadherin patch that we saw towards the center of the aggregate, and the second polarity axis that is on the outer interface of the cell, which describes the inhomogeneous distribution of molecules near the outer cell interface. And then, so we have this interacting active bead model, which is a minimal model for a cell aggregate, much simpler than the one that I've shown before, where each cell is just represented by a sphere, and then we have a way to calculate interacting forces and interacting torques between the beads. In that case, we put two simple rules. One is that when we have this inner polarity axis, if they are close enough to each other, it will create a little spring, which represents the adhesion brought by these E-cadherin patches. And we also assume that the secondary polarity axis creates a propelling force that pushes the cell against it. And then we can solve this model. We take a cell and we make it grow. And this is the order parameter as a function of the active force. So if we have no active force, we only have this inner polarity, and the doublet is dividing, and it makes some structure which has some degree of orientation, maybe kind of hard to see, but which is not really a rosette. There's not really a common cotine point in the center. If we increase this active force, now the cells are moving around, and actually that gives them an opportunity to find a better configuration. And then as long as it's not too high, there is a better order parameter, a better order structure. And if we now remove this inner polarity interaction, but we only have the outer propelling force, then the aggregates become disordered again because the cells are moving, but they don't have a common center point. Okay, sorry, okay. Okay, and so this is my conclusion. So I showed you this model, which are based on polarity interaction. I just wanted to show, to finish this, we can have different flavors of this model, and this is a version where a set of cells assembled in a row can spontaneously assemble into a hollow sphere, and this is a very interesting self-organization process that we can obtain with this model. Okay, and we'd like to thank people involved in this work. Mentioned I have a postdoc position available. Thank you for your attention.